Thank you very much. Any questions or comments, please? Please.
I have three questions. First, when you use the radiation using mice, can we say that cancer patients who are receiving radiotherapy, they should drink deuterium-depleted water to protect the healthy cells?
Yes.
Okay.
Yes, it was. I've seen it. I've seen it done, and it truly reduces the effects of radiotherapy, of cobalt radiotherapy or betatron.
The next question: have you tried to use mice with cancer and make some irradiation?
No, I had no possibility of finding immune-suppressed mice in order to produce.
So we cannot say whether there is some synergistic effect between radiotherapy and cancer. We can say that radiotherapy, so the DDW can protect against radiotherapy, but we cannot say whether there is any. Okay.
I don't see.
The third, of course, this in vitro growth is just the opposite what we are saying. The question is what can be the real reason and the real difference between our two different experiments. One of them, I think, maybe could be the explanation. So when we did this in vitro experiment, we always used the cells synchronized in G or G1 phase, and we checked the first couple of 24, 48 hours, and I also showed that after 12 hours the cells can grow again with the same rate. And have you used that, or have you synchronized the cells before started this experiment, or used?
Well, I think so. I cannot answer you certainly of this. No, let's say no, because if I can't be certain, I don't know. However, the cells were taken from the reserve and were frozen, and then we put them in the exact same time. We put them in to proliferate, to grow. So I think they were synchronized by the force of the. I don't know exactly. I can't respond to you with 100% certainty.
And the question is maybe the genetic background or some other. So maybe some changes must be okay. If we say okay, we found that result that we should inhibit. You found something was different in your system because you say that maybe that stimulate. The time frame is different, I'm sure, because you followed it for several days, these cells. So you cannot say that the effect of deuterium depletion in the first 24 hours.
No.
You say the effect after a couple of days.
24, 48, 72, so until confluence.
Okay.
Until confluence. So and usually confluence is obtained at 4 to 6 days. So at the end of the week, usually we stop the experiment.
Okay. My last comment would be that of course we have to be very worried about the safety, because it's very important not to give something to people which can be dangerous. So I would say that maybe we don't have to worry about, because we have made that type of safety experiment in the phase 2 double-blind clinical trial, and we have lots of experiment with cats and dogs, and after 15 years or 17 years follow-up, we can say that we never saw that somehow that deuterium-depleted water would stimulate the cancer grow and stimulate the metastasis. Of course, we should figure out what's going on in your system.
So this cautioning was made some long time ago, and without being aware of the results of other studies. So.
Thank you. Any other questions, please?
So this is just out of curiosity. Do you know why you cannot really deplete below the 80 ppm in vivo systems?
No idea. So the research was made, let's say, in an unsystematic way. There was no program dedicated to such a research. It could have been better explained if it would have been, let's say, part of a very well-developed program. But this was made, let's say, from time to time during several years, and in, let's say, free slots of time of various connections and friends and things like those. So I cannot tell you exactly why does it happen. The only thing I can tell you is that it happens. Maybe it can go lower, Who knows?
Is it possible that water is produced in our system by various
Mitochondrial metabolism and whatever other
metabolism, and
Krebs cycles?
just because of the ratio of water we consume and the water that is produced from various metabolites just provide this?
The animals received only this water and the so-called exclusively dry food. Well, it's not completely dehydrated. That food is about 25 to 40% also humidity compared, let's say, with 90% other types of food. So there is a certain amount of water, of normal water in the food. And in the air, we have lots of air vapors that we breathe in, and there are probably significant sources of water around us which will not allow the animals to go lower. Maybe it can, maybe it takes more time. It was only 2 weeks, maybe 3 weeks, something like this, 2.5, during which those investigations were made. Maybe if you give it for 6 months or for an year and keep it in a controlled environment, you might decrease it lower. I don't know.
Now, about your research and comparing the rate of proliferation, I'm really fascinated by the various results, and just it's really interesting how actually weight affects various systems, and that is true with sporting activities. In Los Angeles, the Galaxy was actually training with heavy soccer shoes, so when the actual game comes, they will run faster. Now, what was interesting is that really truly they fell in the first few minutes of the game because practically it's just such a different system after training in heavy shoes. Yet later they sped up, so the coach figured out that they cannot really switch shoes from practice to games and vice versa, and there's a little time to adjust to the different conditions. And I think, and I'm going to talk about this later, there is an adaptive effect to light water or deuterium depleted water, which may eventually speed up certain physiological processes. And I definitely think that actually alternating these ratios over time and finding out the appropriate alternating approach in biochemical systems and just knowing the details of how these switches may affect physiology, and that includes cell proliferation and various other physiological processes, eventually there is a scenario we can apply which is biochemically established and clearly understood.
Thank you.